Welcome to Click for IB Biology. We are studying topic D 3.3, homeostasis. We're looking at the functioning of the kidney in osmoregulation and excretion. And in this video, we're going to look at ultrafiltration and selective reabsorption in the proximal convoluted tubule. There's a PDF of this presentation that's linked in down below the video in the description. You can download that and annotate with your own notes. But also then you can follow up with a 10-question quiz of short answer questions to secure your understanding of the topic. Ultrafiltration is a process which takes place in the nephron structure known as the Bowman's capsule. Blood entering the Bowman's capsule enters in the afferent arteriole and is already under high pressure. Blood which exits the Bowman's capsule is in the efferent arteriole, and the important point here is that the lumen, the space through which the blood is flowing, in the efferent blood vessel is narrower. And the effect of this is to create a back pressure in between the afferent and efferent arteriole. This is in the capillary network known as the glomerulus. So in this network of blood vessels inside the Bowman's capsule here, we have the glomerulus and we are under high pressure, much higher pressure. Now, this high pressure, this hydrostatic high pressure, is the force required to bring about the filtration process. But when we go looking for the filtering structure, it turns out not to be the wall of the capillaries, which have gaps in them called fenestrations, capillary fenestrations, and nor in the walls of the Bowman's capsule called podocytes, as they also have gaps. In fact, the filtering structure is a meshwork of protein molecules, fibrous in nature, which create this matrix through which the blood is applied and through which some contents pass and others do not. What passes through is dissolved glucose, amino acids, urea, and salts, and this goes on to form the glomerular filtrate, which is here and here. What does not pass through into the filtrate and remains in the efferent arteriole are plasma proteins, large plasma proteins, and the cells, the red blood cells and the white blood cells. Indeed, if one finds plasma proteins or cells in the filtrate, this is indicative of a number of medical conditions. We'll notice that glucose and amino acids are actually useful substances, and whilst they have been gone through the filtration process, the body wishes to retain these and will therefore select these and then reabsorb them. That's also true of the salts. Urea, on the other hand, will be eliminated. The process that we're now looking at is what is called selective reabsorption, and as the filtrate flows on, it flows into what is called the proximal convoluted tubule. As the filtrate, the glomerular filtrate, makes its way into the proximal convoluted tubule, the selective reabsorption will begin. We can break this down into four main components. Component one is here, which is that glucose and amino acids are completely reabsorbed from the filtrate into the blood. This is a two-step process. Step one is active transport, and step two is diffusion for both glucose and amino acids. Step two is the reabsorption of some particularly small proteins that may have slipped through the basement membrane. This is a process that occurs by what is called pinocytosis, taking these small proteins out of the filtrate here into the cells here. They're digested into amino acids, and then they diffuse into the bloodstream here. The third component, which is reabsorbed in the proximal convoluted tubule, is water. Of the water which is in the renal filtrate at the beginning, more than 80% is actually reabsorbed in the proximal convoluted tubule. And finally, let's consider urea, which is a toxic molecule which needs to be eliminated. And yet, because of its very small size and its lack of a charge, a small amount of urea is actually reabsorbed into the blood. And so this is the significance of urea in our bloodstream, is that it's a small molecule and it has no charge. So it's only a small portion of the urea is reabsorbed into the blood. And so we've got, after the proximal convoluted tubule, the filtrate should contain no glucose, no amino acids. There should be no small proteins, 80%, so the volume of water will be greatly reduced, along with salts actually, and the urea content will be relatively small. We're now going to go on and look at the influence of the loop of Henle on the remaining filtrate. That's the next video, the chance to subscribe to the channel, and the playlist on the right hand side.